Have you ever been told that placing an extra piece of fruit near a banana will make it ripen faster? What's going on there? Well, it turns out that fruit releases a compound called ethylene into the air. And that ethylene acts like a signal telling the banana it's time to ripen. The more ethylene, the stronger the signal. This is a good example of a chemical reaction where the rate is changed by the concentration of things around it. And that's typical. When we run chemical reactions, the higher the concentration of the reactants, the faster it goes. That's what we're going to study in this experiment. We're going to take eggs and we're going to dissolve them in vinegar. Here's the trick though. One egg we'll put in pure water, another egg we'll put in half vinegar, half water, and the last egg we'll put in pure vinegar. And we're going to see what impact this has on the rate of dissolving the eggshell. Let's crack the case on this exciting experiment. Let's begin by reviewing our lesson objectives. First, we'll introduce eggshell reactions and osmosis. Those are two ideas you need to know about to make sense of the lab. Then we'll review the lab procedure, so you know how to do it. And lastly, we'll introduce the required materials for this lab, so you know what you need to get the job done. Let's start by talking about eggshells and vinegar. I'm going to show you a chemical reaction, but don't freak out. I know, it's a little scary. Okay, let's go through part by part. Calcium carbonate, the CaCO3 thing, that's our eggshell. Notice it's solid. And we're going to be mixing that with vinegar. Notice that's aqueous. When you combine our eggshells and our vinegar, the eggshells are going to go away. And what you'll notice in its place is bubbles from carbon dioxide gas. So those are the important things you need to keep in mind for our eggshells and vinegar reaction. Let's move on to osmosis. Remember that water flows to equalize ion concentrations. Let me explain what that means with a few examples. Here we see a red blood cell, and this red blood cell has salt ions on the inside and the outside. On the outside, the ions are represented by those light blue spheres, and on the inside, the ions are represented by the dark blue spheres. Notice that there's a low concentration of ions on the outside, so not that many ions, whereas there's a bunch of ions on the inside of the cell. So what happens then is water flows from the outside to the inside, diluting that high concentration of ions, trying to equalize the concentration. So water flows towards higher concentrations. The opposite is happening here. Notice that on the outside, we have a high concentration of ions, while on the inside of our shriveled cell, we have a low concentration of ions. So the reason that cell is shriveled is because water is flowing out of it. So the cell shrivels as water leaves. All right, well, now let's review our required materials. We'll need 3 24-ounce cups. Mason jars work really well here, but any large cups will do. We're going to fill the first cup with pure water, and the second cup with half water, half vinegar, and the last cup with all vinegar. To measure the water in the vinegar, you need a 1-cup measure. Into each of those glasses then we'll place an egg, so 1 egg per glass, and we'll cover it with plastic wrap. Lastly, you're going to need time for this experiment. This experiment must sit for 24 hours, so make sure you plan ahead to have a little time to let that rest before you come back and take a look at these eggs. Here are all your required materials. If you're ready to begin, go ahead and gather these required materials, and then you can continue the video lesson. All right, well, lastly, let's go over our lab procedure. First, let's label the cups water, 1/2 vinegar, and vinegar. And then you'll fill the cups according to the instructions. Basically, 1 glass will be pure water, 1 glass will be half vinegar, half water, and the last glass will be all vinegar. You're going to add 1 egg to each jar and record observations. So you want to make sure and carefully record those observations before you let them sit for a while. Then, 24 hours later, remove the eggs and record your observations. Now you might be wondering, what observations are we going to be making? Well, here are some guidelines for your observations. First of all, compare the quantity of bubbles produced by each egg. This is your initial observation tips. Also record if the egg sinks or float, and lastly, record the color of the eggs. Then, 24 hours later, you're going to make your final observations, and once again, you'll record if the egg sinks or float. You'll compare the quantity of eggshell remaining, so that's going to be a new type of observation, and you'll compare the size of the eggs. Before you get started, make a hypothesis. Remember, that's just a fancy word for a guess. Which eggshell will dissolve the most or the least, and why? Will the size of each egg increase, decrease, or stay the same? Why? And then, this is the fun part, compare your hypothesis to your observations at the end. All right, well, let's review what we've learned. First, we introduced eggshell reactions and osmosis. We saw that our eggshell is going to dissolve in vinegar and release carbon dioxide, which we'll see as bubbles. We also saw that osmosis allows water to flow in and out of things, that water always flows towards the higher ion concentration. Then we reviewed the lab procedure. Basically, we got 3 jars. 1 jar has pure water, 1 jar half vinegar, and the last jar all vinegar, and we're going to plop an egg in each one. Then we introduced the required materials for this lab. Hey!